Ever wonder how a thought flashes through your mind or how you instantly feel the sun's warmth? It's all thanks to neurons, the brain's tiny messengers. These specialized cells are the wiring of your body's supercomputer, carrying information, processing thoughts, and controlling movement. Without neurons, we couldn't think, feel, or act. Picture a neuron as a tiny tree. Dendrites receive signals, the cell body keeps things running, and the axon sends messages onward. Billions of these cells form a vast, interconnected network, passing signals at lightning speed. The real magic happens at the cell membrane, a thin, active barrier that separates the inside and outside of the neuron. This membrane controls the flow of charged particles, or ions, like sodium and potassium. By managing these ions, the neuron stores and releases electrical energy, its essential signal. This creates a small electrical difference, called the resting membrane potential, like a tiny battery ready to fire. Every thought and sensation starts with this electrical property. It's the quiet before the storm, a charged cell, waiting for action. Understanding neurons is the first step to understanding ourselves. Let's see how this electrical storm begins. Imagine a hydroelectric dam holding back water, full of potential energy. A neuron's membrane works the same way, but with ions instead of water. The cell pumps potassium inside and keeps sodium outside, creating an imbalance. This separation builds up electrical potential, like a dam ready to release water. The membrane voltage is this stored energy, always maintained by molecular pumps. The neuron is primed, like a coiled spring, ready to fire at a moment's notice. The membrane acts as the dam wall, storing charge, a property called capacitance. All it takes is a trigger, a signal from another neuron to open the gates. When that happens, the stored energy is unleashed in a burst of electrical activity. This burst is the neuron's signal. The readiness of this system is why your brain can react in milliseconds. The stage is set for the action potential. Let's see what happens when the floodgates open. The electrical storm is about to begin. When the floodgates open, we get the action potential, the nerve impulse that zips down the axon. It's all or nothing. The neuron either fires or it doesn't, like flipping a switch. It starts when enough stimulation pushes the membrane voltage to a threshold. Voltage-sensitive sodium channels open and sodium rushes in, making the inside positive. This is depolarization. The voltage spikes upward, but quickly, sodium channels close and potassium channels open. Potassium rushes out, bringing the voltage back down. Repolarization. The membrane briefly dips, even more negative than usual, hyperpolarization, before returning to rest. All of this happens in milliseconds. The neuron resets, ready to fire again. This rapid wave of electrical change is how neurons send signals. Every thought, movement, and sensation depends on this process. The action potential is the electrical language of the brain. But what makes these ions move in the first place? Let's look at the forces behind the flow. The story of the action potential is a story of moving ions. Why do ions like sodium and potassium rush in and out of neurons? Two forces drive them: diffusion and electricity. Diffusion is the tendency for particles to spread from high to low concentration, like food coloring dispersing in water. In neurons, potassium is concentrated inside, so diffusion pushes it out. Sodium is concentrated outside, so diffusion pushes it in. The second force is electrical. Opposite charges attract, like charges repel. The inside of a resting neuron is negative, pulling positive ions inward. For potassium, diffusion pushes out, electricity pulls in. For sodium, both forces push it in. The balance of these forces, the driving force, determines ion flow. The action potential is a rapid shift in which force dominates for each ion. This invisible tug-of-war is the engine of neural signaling. Next, let's see what happens when these forces reach a perfect balance. That's the equilibrium potential. Is there a point where diffusion and electrical forces on an ion cancel out? Yes, the equilibrium potential. For potassium, diffusion pushes it out, but as the inside gets more negative, electricity pulls it back in. At about 90 millivolts, these forces balance. For sodium, diffusion and electricity both push it in, but as the inside gets more positive, the electrical force pushes back. Balance is reached at about plus 160 millivolts. At each ion's equilibrium potential, there's no net movement of that ion. The membrane voltage at any moment depends on which channels are open. At rest, the neuron is mostly permeable to potassium, so its voltage is near potassium's equilibrium. During an action potential, sodium channels open, and the voltage races towards sodium's equilibrium. The dance of the membrane voltage is a story of shifting permeability. Now, let's see how neurons control this flow. Electricity in neurons follows rules, just like in wires. Ohm's law tells us current equals voltage divided by resistance, or, in neurons, conductance times driving force. Conductance measures how easily ions flow. It depends on how many channels are open. The driving force is the difference between the membrane voltage and an ion's equilibrium potential. If the voltage is far from equilibrium, the driving force is strong. When sodium channels open at the start of an action potential, conductance and driving force are both high, so sodium rushes in. The neuron controls signals by opening and closing channels, changing conductance. If channels are closed, no current flows, no matter the driving force. It's like a dam. Water can't flow if the gates are shut. The precise timing of channel openings is how neurons send rapid, reliable signals. Next, let's see how these channels actually work. The secret is in their gates. Enter the world of ion channels. Ion channels are complex proteins with gates that open or close in response to voltage, voltage-gated channels. Hodgkin-Huxley modeled these channels with gating variables. Potassium channels have 4 identical gates. All must be open for potassium to flow. The probability is nn to the 4th. Sodium channels are trickier. They have 3 activation gates, M, and 1 inactivation gate, H. All 3 M gates must be open, and the H gate must also be open for sodium to pass. So, conductance is M cubed times H. At rest, M gates are closed, and H is open. When voltage rises, M gates open fast, H closes slowly. This explains why sodium current turns on quickly, then shuts off even if the voltage stays high. The interplay of these gates creates the action potential's rapid rise and fall. This elegant model matches real neuron behavior. The opening and closing of these gates is the key to neural signaling. Hodgkin and Huxley's insight changed neuroscience forever. Let's see how they put it all together. The neural code was about to be cracked. Hodgkin and Huxley combined membrane capacitance, equilibrium potentials, and gating variables into a set of equations, the Hodgkin-Huxley model. This model describes how the membrane voltage and gating variables change over time, predicting the action potential shape and behavior. Their experiments on squid axons let them measure currents and fit their equations to real data. When they ran the model, it matched the real action potential perfectly. The model could predict thresholds, spikes, and responses to stimuli. It became the foundation of computational neuroscience. Decades later, the actual structure of ion channels matched their predictions. The Hodgkin-Huxley model showed that the brain's electrical signals could be explained by physics and math. It was a triumph of theory and experiment. But what does this mean for us? The Hodgkin-Huxley model began with a squid neuron, but its principles apply across the brain's billions of cells. Real neurons are more complex, with many types of ion channels and intricate shapes, yet the model provides a foundation for building more detailed simulations of brain activity. Understanding these electrical signals is crucial for medicine. Many brain disorders involve faulty signaling. Epilepsy, for example, is caused by neurons firing uncontrollably. Targeted drugs can restore balance by acting on specific channels. Decoding the action potential is the first step to treating these conditions. Ultimately, understanding neural signals helps us tackle the biggest questions. How do we think? What is consciousness? Hodgkin and Huxley gave us the alphabet of the brain's language. By learning to read it, we move closer to understanding ourselves. The journey to unlock the mind begins with a single neuron, and that's the true wonder of neuroscience.